our cells, the cells actually go through exocytosis more often than they go through endocytosis. So over the lifetime of cells, they get bigger. And eventually what will happen is the cell will get to a certain genetic large size, and then that cell will divide. It will go through mitosis, and then it will become two smaller daughter cells. One of the problems is sometimes our DNA doesn't work quite right and doesn't allow that cell to actually go through this mitosis. And so instead, the cell just keeps getting bigger and bigger and bigger. Now, the way our cells are situated, okay, you've got these cells sitting next to each other on top of other cells, and these cells are held together by these proteins that kind of sew the cells together. But if we have a cell that gets really, really large, so this cell is going to get a lot bigger than it's supposed to be, it becomes so large, it's so heavy, that eventually it will rip these little protein stitches that hold it to the other cells. Now this cell that doesn't have the right genetic information can actually break off of the other bundle of cells and move to a different area of the body and settle there. And when it does, it actually can release particular chemicals that cause other cells to not be able to go through mitosis appropriately, and they get larger and larger and larger, and they begin to lose their function when they get too large. And this is metastasizing cancer. So any cell that gets too big and can't go through mitosis, loses its original function, and then starts floating to other places, can then stimulate cells that should have never had a problem start to go through this kind of unusual process and become cancer forming. So our cells should only go through so many rounds of exocytosis before they divide and become smaller again, because those are the cells that are going to stay healthy. This is just a picture showing you this electron micrograph of a cell going through exocytosis, and you can see all the proteins that it's releasing at one time as the vesicle joins here with the cell membrane and makes the cell a lot larger. Any questions? No? We're good? Okay. So we're going to go on to the next PowerPoint, which is all about cells and tissues. We've been talking about the cell membrane, and just a couple more things I want to go over before we actually get into the organelles of the cell. And that is that sometimes the cell membrane isn't just flat. You've got projections that come out of the cell membrane. So for instance, in this picture here, we're looking at cilia. So this is an electron micrograph that blows up the cell membrane about 250,000 times its size. And you can see the cilia, their little finger-like projections that come off the membrane. They have protein that goes up inside of them. And you know, we've talked about this, that protein has the ability to change shape. Now we have the same thing in other instances. Besides cilia, we have microvilli, we have flagella, which would be the tail at the end of the sperm. These all have protein in them as well. And these are the proteins, for instance, inside the cilia. These proteins, because they have the ability to change shape, the cilia can bend. And so we have cilia, for instance, in our trachea, okay? And that cilia is constantly bending and coming back to its original shape and bending again. And as it bends, it's pushing, for instance, in the trachea, all of the mucus and dust particles, anything we inhale that we aren't supposed to, up and out, so it doesn't get down into our lungs. And then we have the ability to keep the lungs clean. In the female reproductive system, we also have cilia, for instance, in the fallopian tubes. And that cilia helps to move the egg from the ovaries to the uterus. So cilia is very important in helping us to move particular substances, and of course this is found on the surface of the cell membrane. So far we have talked about the cell membrane. We're going to talk now about the organelles within the cell, and then after that we'll go into the nucleus of the cell, and then it's time for your first exam. So inside the cell we have what's called the cytoplasm, and the cytoplasm is basically made up of two parts. We have the cytosol, which is the watery portion of it, and then we have the organelles. Now the cytosol, it is similar to your agar, not quite as thick. It's what we would call a semi-gelatinous substance, so maybe more like the thickness of egg whites, okay? But it's not so thick that your organelles can't move inside this cell. They're able to move around. Things aren't stagnant. Things are able to move through the cytoplasm. And so in this cytoplasm, we'll have the organelles, and then we'll have a whole bunch of other chemicals too. You'll have ions and glucose, all kinds of different types of nutrients, enzymes, waste products, all kinds of things are moving inside this cell. Now our organelles, they're going to be the worker bees, you might say, for the cell. They're the things that are helping us to make various products inside of our cell. Some of these organelles, as a matter of fact, most of the organelles have a membrane around them. There's a few that do not. The membrane is pretty much the same as the cell membrane. Phospholipid bilayer, integral proteins, everything that we talked about in a cell membrane would be the membrane of an organelle. Excuse me. Now, one of the organelles that does not have a membrane would be our centrioles. Okay, so let me just erase this real quick, and we'll talk about what the centrioles are going to do for our cells. So in our cell, we typically only have one pair of centrioles, unless the cell is going to go through mitosis. So if our cell is going to divide, you're going to see two of these centrioles inside the cell. And basically they're going to be located at, you might say, like the North Pole and South Pole of the cell. And these centrioles, they kind of remind me of, like, you know, Spider-Man. They're able to produce these long strands of proteins that go between each of the centrioles. So they're like, you know, shooting out the proteins like Spider-Man does. And those proteins that are like clotheslines between the centrioles are where all of the DNA line up. So do you remember in anatomy when you had to look at all those slides, you know, and you had to look at like prophase and metaphase and anaphase? And if you remember, all the DNA lined up right down the middle of the cell in metaphase, and then those DNA parts got pulled into both of the cells. It's the centrioles that are pulling the DNA and pulling the two cells apart. If you don't have these centrioles, you can't pull your two cells apart in the process of mitosis. Organelle is what we would call the rough endoplasmic reticulum. So let's go back to this picture real fast here. And notice right here in the blue, this really large organelle, this is the rough endoplasmic reticulum. It's the largest organelle in the cell. It attaches to the cell membrane. It also attaches to the nucleus of the cell. And we'll talk about why that's important a little bit later on when we talk about protein production. So this right here would represent the rough endoplasmic reticulum, and this is the membrane of our nucleus. Now, when scientists were looking at the cell and trying to figure out what the organelles were, what they did, all that kind of stuff, somebody took one look at this rough endoplasmic reticulum and they said, okay, well, it's got all these little dots or bumps on it, and so we'll refer to this as the rough endoplasmic reticulum. And then they found another one inside the cell that didn't have all the dots and bumps on it, so they called it the smooth endoplasmic reticulum, okay? Well, it turns out the dots and bumps on the rough endoplasmic reticulum is this guy right here. This is a ribosome. So all these little dots was actually another organelle, which is the ribosome. So here's a picture of a ribosome. Ribosomes actually can come apart. There's two parts to them. And when they come apart, one of the parts is a little bit bigger than the other. So they call it a large subunit and a small subunit. And ribosomes, they're really important in the cell's ability to make proteins. And so when we talk about protein production in a couple days, we'll talk about what exactly the ribosome is doing, how it actually helps to make these proteins. So again, the rough endoplasmic reticulum, you can see this here. This is what they call a transmission electron micrograph. And all these little dots, these are ribosomes on the rough endoplasmic reticulum. Now, the rough ER has several jobs. One of those jobs is it also assists in protein production. But another really, really important job for the rough endoplasmic reticulum is it stores calcium. Every single cell in our body has a rough endoplasmic reticulum. And every single cell needs it because we use calcium all day long. Constantly our cells are using calcium for a lot of different chemical reactions. So we have to have a really good storage area for calcium. Now, we also have the smooth endoplasmic reticulum. Like the rough endoplasmic reticulum, it also stores calcium. But then it also has a lot to do with fat, not proteins like the rough. The smooth is all about fats. It produces fatty acids and steroids and phospholipids. So the various different types of fats we've talked about in class, the fatty acids for our triglycerides, our steroid hormones, and our phospholipids for the cell membrane. These are all produced in the smooth endoplasmic reticulum. The next organelle is called the Golgi Apparatus. It has two jobs. One is called glycosylation, and the second is vesiculation. Now, we already talked about this today. I just didn't use those terms yet. But the Golgi apparatus is assisting with exocytosis, where proteins are leaving the cell. So now, here you go, ma'am. And I think she's still in there, so you can sign it and just give it right to her. Thank you. So now, the Golgi apparatus, the first thing it's going to do for a protein that is produced inside our cell is it's going to put glycogen on it. So we're going to take this protein that we need to ship out of the cell, and we're going to add that glycogen onto the protein. Now the reason that we add glycogen onto a protein that's being shipped out is because, remember, this glycogen has a very particular pattern, and that pattern belongs to you. And when we ship that protein out of the cell, if an immune system cell sees it, it will not destroy it. Otherwise, if you don't put glycogen on it, or if you don't glycosylate the protein, there's potential for our immune system to destroy what we just made. And then, you know, anytime we ship a protein out of the cell by exocytosis, we have to put this protein into a vesicle in order for that vesicle to move to the cell membrane and fuse with it and release that protein from the cell by exocytosis. Putting a protein in a vesicle so that exocytosis can occur is what we would call vesiculation. So glycosylation, we add a glycogen to it, and vesiculation, we put it in a vesicle so it can leave by exocytosis. And those are the two jobs of the Golgi apparatus. So if you look at the overhead, you'll see here these little round things that are coming off here at the top. These are the vesicles, like the little bubbles that are going to head to the cell membrane, and whatever is inside, whichever kind of protein, it'll be released by exocytosis. Basically same thing here. You can see they've opened the vesicles up so you can see the proteins that are being transported. And then here's the exocytosis occurring. Any questions about anything so far? All right, so the next one is the lysosome. Digestive enzymes. Probably, depending on what lysosome we're talking about and which cell, anywhere from like 50 to 100 different types of digestive enzymes. So now remember, we talked a little bit earlier about phagocytes. And phagocytes take their little arms and they grab a hold of a bacteria and they bring that bacteria in. Once that bacteria comes in, it's going to go to the lysosome. So look at the overhead. Here's our phagocyte, and it's trying to eat up this bacteria here. The bacteria gets inside, and it is transported into a lysosome. The lysosome will then digest that bacteria, and then notice what gets exported out, just a little teeny tiny bit of waste product. Or, depending again on the cell, the lysosome might keep that waste product in the cell. So sometimes lysosomes will build up waste products, which could be the reason that some of our cells end up dying. Because they build up so much waste product, it ends up destroying the cell. You can't have too much waste product building up inside cells because there's not enough room for things to occur. Also, these waste products always, the majority of the time, have an acidic pH, and you get too much waste product inside the cell, and that acid can then also destroy the cell. Which, by the way, is what a lot of people think happens like with rheumatoid arthritis. The lysosomes are creating too much waste product. Those waste products stay inside the cell and build up, and then they begin to destroy the cells, especially around the joints, and you have rheumatoid arthritis. This can also be a type of autoimmune problem where the phagocytes are releasing digestive enzymes from their lysosomes onto other cells and causing those cells to die, and then you have RA, which is not a fun disease for people to have. The next organelle is a peroxisome. Peroxisomes secrete an enzyme called catalase. Now, the job of catalase, this enzyme breaks apart H2O2, this is hydrogen peroxide, and breaks it apart into water and a single oxygen atom. 1/2 O2 is the same as just O. The 1/2 and the 2 cancel each other out. I don't know why we can't just write O, but I'm not a chemistry person, so this is how they decided to do it. Now, the peroxisome only secretes one enzyme, and this means it only destroys one waste product. It doesn't do anything else, which is really interesting that you have a single organelle that is there to digest and destroy a single waste product. Seems sort of like a waste of energy to me, unless that waste product really needs to be destroyed. And you really need to be able to get rid of that thing out of the cell. So why is it so important for our cells to destroy something like hydrogen peroxide? Well, let's talk about hydrogen peroxide like you can get at the, Rite Aid or whatever. So, you ever bought hydrogen peroxide? What color bottle does it come in? Brown, absolutely. And the reason they put it in a brown bottle is because ultraviolet light affects hydrogen peroxide. Now, what is the percentage of hydrogen peroxide in that bottle? Do you remember?
1%.
It's about 2%, which seems like a ripoff. That's like 98% water, you're only paying for 2% hydrogen peroxide. But here's the reason why. If you had more than 2% hydrogen peroxide, like let's say you bought a bottle that was 50% hydrogen peroxide and you picked it up off the shelf, it would blow off your arm. I think she's still there. Okay. So it would literally blow off your arm because hydrogen peroxide is explosive. And by the way, we put it in a brown bottle. They keep it in that bottle because in that bottle, if it hit ultraviolet light even at 2%, it would bubble. And then when you poured it on your wound, there wouldn't be any bubbling. It would already be bubbled out. So you have to keep it in the brown container, and you have to keep it at a very low percentage, or it's like explosive like dynamite. You could like see all these Rite Aids blowing up everywhere. It'd be really fun, kind of fun, but anyway. So then, why do we pour hydrogen peroxide on a wound? What is it doing? Lysing what? I hope not. That would be really nasty. What is it supposed to do? It cleans things out. It kills bacteria. So you're pouring it on. You see all those bubbles, right? Well, those bubbles are like little bombs to bacteria. It's literally causing those bacteria, not your red blood cells, but the bacteria to actually go through lysis. Now, do we use hydrogen peroxide in the hospitals to clean wounds? No. No, 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 never. It's never used. And the reason for that is because if you were to take hydrogen peroxide and pour it on a wound day after day, it would then begin to lyse your own cells. Not just the bacteria that's there, but your own cells would start to explode as well. That would not be good, and the wound would get bigger and bigger and bigger. I actually had a student. Sometimes I don't get very bright students, and I don't know how they get to my class. It's like I want to yell at the other teachers for, how did you let this idiot get into my class, okay? And he decided that he had a little wound on his hand, and he decided to see if I was lying or not. Okay? And so he started pouring hydrogen peroxide on his wound. And he did it like day after day. And he came in one day and he's like, Dr. Harvey, you were so right. Look at my hand. And I swear to you, I could see the bone.
Oh my God.
And I'm like, yeah, I don't need you to tell me that I'm right. I know I'm right. Get your butt to a hospital right now. I mean, he had to go through skin grafts and everything to bring his hand back together. He also dropped out of my class, but, do not do this at home, okay? This is what I'm trying to tell you. If you don't believe me, instead of trying it on yourself, look it up. That would be probably anyway. Some people do some interesting things, I don't know. But now, think about this for a second. Could you imagine if there was a person who did not have peroxisomes? Let's say maybe they had some kind of genetic defect and they didn't have any peroxisomes in any of the cells of their body. What would happen to them? They'd explode. How cool is that? You know that spontaneous human combustion thing? That was so cool. I love it. Unfortunately, there's no scientific proof that nobody has or anybody has no peroxisomes. Everybody seems to have them. Nobody's exploding, so bummer. All right, the next organelle is the mitochondria. The mitochondria produce ATP. At least most of it. We can produce some ATP in other places in the cell, but most of our ATP that we need to change the shape of enzymes is produced in the mitochondria. Now there's one other thing I want you to know about mitochondria, which it's really kind of interesting, is mitochondria has what we would call mitochondrial DNA. Now anytime you're reading about DNA, you may see it written as mtDNA, and the MT stands for mitochondrial. Or you may see it just written as DNA, or you may see it written with a little N in front of the DNA. If it's just DNA, or it's nDNA, then know that that's the DNA inside our nucleus. The mtDNA is DNA found inside the mitochondria. Now, mitochondrial DNA gives the mitochondria the ability to do something that no other organelle has the ability to do. The mitochondria have the ability to replicate. They can duplicate themselves. They can make more mitochondria. Why would you want to make more mitochondria?
More ATP.
More ATP. And why would that be beneficial? Why do you want to make more ATP? Because you speed up your enzymes and you would feel good. It's kind of like taking Adderall or Red Bull. You'd have a lot of energy. The more mitochondria you have, the more energetic you feel. So usually somebody says about this time, well, how can I make some more mitochondria? Because I'd really, really like to have more energy. And I'm sorry, I'm going to have to tell you something that you're not going to want to hear. The only way we can produce more mitochondria is through exercise. I know that's like illegal in the United States, okay? And not something that we're supposed to do. But it is the only way that our cells are willing to make more mitochondria is the more exercise we do, of course, the more energy we need, and the more mitochondria we're able to make. Now, here's the other question. How much exercise do I have to do? How long do I have to do it for before I can, make some more mitochondria? So, let's say you have somebody who hasn't been exercising for, I don't know, like the past 10 years. They've been busy with their career, haven't had time for this, no real exercise. They just go to work, come home, sort of veg on the couch, and go to sleep, okay? And then they decide, okay, that's it. I am going to go out. I am going to exercise. I'm going to put my running shoes on. And I'm going to go out the door. I'm going to take a run. Now, after 10 years of doing nothing, except going to work and coming home, how far do you think this person's going to get?
Not very far.
Yeah, not very far. Probably like, quarter mile down the road and is like on the cell phone, honey, come pick me up. I don't think I can make it home type of thing, okay? Now, I'll tell you though, while he was running, his mitochondria were having a little conversation with each other going, he's moving. This is very unusual. I don't quite get it. Should we start duplicating ourselves? And the rest of them were like, no, this is not going to happen again, trust me. Okay, but you know what? No, this guy is really dedicated. He is done living this slacker life. He is going to get himself in shape, puts his little running shoes on, and he is ready to go again. Now, if he does this, let's say maybe 2 days later, how far is he going to get? Probably just about the same, and he's probably still going to need a ride home because the mitochondria haven't made any more ATP. They haven't replicated. He doesn't have any more energy than he had before. As a matter of fact, he probably has to wait 2 or 3 days since the last time he exercised because he used up so much of his ATP, he just didn't have enough to get himself up to do it the next day. So, how long is it going to take this guy who hasn't been exercising for like 10 years before his mitochondria start taking him serious and begin to make a couple more mitochondria for a little bit more DNA? How long is he going to have to do it if he does it every other day? About 2 weeks. He's going to have to do it for about 2 weeks. And after about 2 weeks, when he gets out to go run, one of the things he's going to find is he'll feel a little bit better, a little bit more energetic, maybe be able to go a little bit further, or instead of asking for a ride home, he can walk home. But it takes about 2 weeks. And you got to remember that because your patients, they don't want to exercise any more than you do probably. And you don't want them to do it every day. You want them to do it every other day so they have a little bit of time to heal. And you want to tell them, look, you're just not going to feel quite good for about 2 weeks before you start building up some energy. Now, what if this guy is really dedicated and he gets out there like every other day and he's running every other day? How long is it going to be for him before he's actually going, wow, I feel pretty good. I'm kind of energetic as I do this. I've gotten a lot further. I'm seeing massive changes in my body. I feel more healthy. How long before he notices all of this? Yeah, that's the part that really sucks. It's about 6 months. Now, if you have a person who hasn't waited for 10 years to get up and exercise, it's probably going to be a shorter time than that. You're probably going to see somebody feel better in about 3 or 4 months. If you have somebody who hasn't been exercising for even longer, let's say they haven't exercised for 20 years, it'll still be about 6 months. It's kind of the interesting thing. So they'll still see a really significant change in their body if they can do this for a 6-month period. After about 1 year, if they can keep this up and they're exercising, they're actually going to feel, like we talked about before, that person who goes out and exercises and they feel better after the fact than they did even before. But it takes about a year of a continuous type of exercise to be able to feel that if you've been slacking for a long period of time, if you haven't exercised for quite some time. Any questions about this? Hang in there, we're almost done. Okay. Last concept I want to talk to you about before I let you go for the day. This is a special type of cell. This type of cell is called a mesenchymal cell, or it's also called an undifferentiated cell. Now people look at this and go, mesenchymal cell, undifferentiated cell, I don't quite get this. So when these became very popular in the media actually changed their name and called these stem cells. So biologically, we call them a mesenchymal cell or an undifferentiated cell. But when you read about them, like in magazines and newspapers, we call them a stem cell. Okay, now, what is a stem cell? So you have this egg and you have this sperm. And they get together and you make a fertilized cell. That fertilized cell is a stem cell. Now that cell doesn't look like any human you would know. It just looks like a cell. Now eventually that cell's going to divide and divide and divide and divide, and you're going to end up like with a ball of 100 plus cells. And that ball of cells still doesn't look like a human. And every single one of those cells is a stem cell. Eventually, what's going to happen is in that ball of cells, a group of those cells are somehow going to be given some kind of chemical signal, some kind of signal, and that group of cells are going to turn into, let's say, the brain and spinal cord. And then there's another group of cells, let's say, turn into the cardiovascular system. And another group of cells turn into the kidneys. When those undifferentiated or those stem cells turn into something we would recognize as like part of the human body, we say that they are now differentiated cells. So a differentiated cell would be like a heart cell, a skin cell, a muscle cell, a cell that we would recognize as part of a human. But if this cell has not turned into anything, it is an undifferentiated cell. Now, many years ago, probably around 50 years ago, scientists were able to figure out that our bodies had things called stem cells. So in your body and my body right now, you have a lot of stem cells still. Even though you're a fully developed human, you actually keep some stem cells and you're able to make some stem cells in your body. And so scientists were like, oh, this is really cool because these cells could actually turn into any cell in the human body if they were just given the right instructions. So in the very beginning, the scientists were able to isolate some of these stem cells, and one scientist in, I believe it's Switzerland, he started injecting stem cells into people's knees. People who had had like knee injuries, they had arthritis in the knees. He thought, let me just try injecting these cells right into their knee and see if those cells would turn into like cartilage or bone and help to repair. And you know, they did. They were able to turn into, at least most of them, into cartilage and help to repair knees. These people's knees became better. That was just like brilliant news all over the world that this doctor was able to do that. Another researcher who actually worked at Loma Linda at the time decided he was going to try stem cells in his Parkinson's patients. Now, in our brain stem, there is a small area called the substantia nigra. And in Latin, the substantia nigra for us means the black spot. And there's actually a little black spot in the brainstem. And that black spot, it controls our smooth movement of our limbs. So, you know, if you want to move your hands or your arms, they move very smoothly. A person with Parkinson's disease, if they wanted to move their hands, they move it, you know, kind of like this. They don't have smooth movement because the substantia nigra isn't working correctly. So what this doctor at Loma Linda did is he just took some stem cells and injected it right into these people's brain stems, right into their substantia nigra. And it improved their Parkinson's dramatically. Didn't cure it, but improved it. And these were some of the very first research projects done with stem cells. And so obviously you can imagine scientists were very excited about all of this. And so what they started to do was try to grow and control stem cells in a petri dish. Because could you imagine, you put some stem cells in a petri dish, you give it the right information, and they all grow into a heart. And then you could use it to give somebody a heart transplant. Or you give it the right information, and it grows into a kidney. And so they've been working with stem cells for a number of years, and it is really slow going because it's hard to figure out. First of all, stem cells didn't like living in a petri dish. That was the very first problem. As soon as they got into a petri dish, it was like, ah! And they would all kind of crinkle up and ball up, and they just wouldn't stick to the petri dish. And it took about 20 years for scientists to figure out how to just get them to stick. let alone to grow. And they're still very much working on trying to figure out how to use these stem cells. I don't know if you saw it, but just a few years ago, there was a big article out where they were able to use stem cells to grow an artificial ear. And they actually took a little mold and they grew stem cells around this ear, and then they took that ear off of the mold and they did an ear transplant for somebody who was a burn patient. And they put a new ear on. And they got the stem cells from the person's own body, which then of course had their own MHC on it, and their body did not reject any of that transplant. And like I said, you and I still have stem cells, so wouldn't it be awesome if, let's say, you know, I needed a heart transplant, they could take some of my stem cells and grow a heart just for me, and then I wouldn't have to worry about immunosuppressant therapy or getting cancer from that or anything. I just have my own heart. I wouldn't have to wait for somebody else to die and get the heart from them. They'd be able to grow all that. Now, one of the big controversies about stem cell research is that aborted fetuses obviously have tons of stem cells. And so a lot of scientists use an aborted fetus as their stem cell farm, you might say. They're getting their stem cells from the fetus. And a lot of people are turned off by this because what you do is you take that aborted fetus and you put it in a blender. Okay, just like the blender you'd make a milkshake in. And you drop it in a blender and you blend that up, and then you sieve out the stem cells and you separate them from everything else, and then you grow those. And of course, a lot of people very much are opposed to that. Other researchers use stem cells from umbilical cords. So when a baby is born, the only thing inside an umbilical cord is basically stem cells. That thing is just packed full of them. So if you take the umbilical cord, you can use those stem cells for research. Do you have a question for me?
Are you okay with me to try it?
Yeah. Yeah. No, you're fine. Yeah. So also, I don't know if any of you have had children, in the past couple of years, but probably one of the things they may have asked you is, do you want us to freeze the umbilical cord? Do we want us to save that for you? And the whole idea is you're saving your child's stem cells so that in case in the future that we're able to use these stem cells, they have their own store of them. It's probably a little expensive to do that. I heard it's like $150 a month. I don't know if kids are worth that. No, but even as an adult, we have lots of stem cells, so you could still donate your own stem cells, later on in life, even if you didn't have an umbilical cord stashed away somewhere. But it's very, very interesting research, obviously. Any questions?